everyone. How are you? It's 8 a.m. Thank you so much for coming. I know this is a hard time for lectures, but yeah, I appreciate that you're coming and your effort. I mistakenly said last lecture that clicker start counting today. We will follow the syllabus always. And so the syllabus states that clicker starts counting next week, week 3, and that's what we will do. However, we will continue testing the clicker. So this is the frequency is AA, the frequency that we will use today. We may change next week, but I'll let you know. Okay, and I think there were some, I didn't realize it didn't show there. There were some clicker questions that we didn't finalize last lecture, so we will start with those ones. And for some reason, this is not showing there, so I thought it was. Let's see. Oh. Okay, what is it? Okay, there you go. Okay. Okay, there you go. So there are, again, we will start quickly again with the clicker questions we didn't finish. So we are done with all the clicker questions from lecture 2, and then we will do a summary for the lecture 3 that you have already listened to the pre-recorded. And of course, you get the most when you ask your questions, okay? And so I'm always happy to get your questions, and I'll do my best to answer. And then we will do clicker questions of lecture 3, okay? So let's finalize with these ones. They were pretty easy, and but of course, feel free to interrupt and ask questions. So any question before we start? Yes. AA doesn't work? Oh. Okay. The app or the, but because I still didn't pull, maybe, is it working now? No? It doesn't, okay, let me see. I'm not detecting any response, so you are right that it doesn't work. And I did exactly the same as last time, so let's see. This is, there I started to detect responses. Okay, so who is able to connect through the clicker? And you have an app or physical app? The app. And can you try another frequency just in case? You try AA. Can you try AB or AC just in case? AC? AC started working. No? It doesn't work for you? Maybe, okay. Let me see if I can change this to AC, and I can do some changes here. I'm sorry about that. Again, you have the clicker questions in the canvas. If you can continue with that on your own, let me resolve this. it was, yeah, I don't know, I didn't expect this. Can you check there if that brings you to it helps at all when you scan? Okay, I will put AC. Okay. Does it work now with AC? Yes, okay, there you go, okay. Okay, let's do this one first, and the poll has already started, so I give you another 20 seconds. Have we done this one last lecture? Okay, sorry. So the B is the correct one, right? Like the opsonization of pathogens and killing. So I'll stop this one now. And then can you remind me a little bit? So we did this one. Did we do this one? Maybe Job, you know where, I don't know, I don't see Job now. Eric, do you know where we stopped last lecture? Do you guys want to still do them again? Yes? Okay. So we'll go ahead with this one then. The poll is started. This is tricky. Okay, let's see what you answered. Okay, most of you answer C, and I think it's the correct one, but there is something that I want to discuss because I don't know if it is correct. Okay, let's stop it. And so this is, let's see if I can today show your answers. Okay. Anyway, so most of you answer C, which is correct. That's for sure, right? Like C3b is an opsonin, and then it will lead to the endocytosis of the bacteria. It will facilitate the uptake of the bacteria by the macrophage. Most typically, that will lead to the formation of the phagolysosome, right? Like most, I mean, not guarantee, because some pathogens like Mycobacteria tuberculosis or Mycobacteria leprae, they can interfere with the formation of the phagolysosome. So I guess that is why makes C the only true one, because the formation of the lysosome may be subverted by the microbe, but right after the opsonin a binding to the pathogen, there will be the endocytosis of the bacteria. This is definitely true, okay? I think that's the reason, and yeah, I'll go there. Release of enzymes. The enzymes are already there in the lysosome, right? And so you don't need to release them. You don't need to the macrophage doesn't need to synthesize them again. So I think that this is wrong because of that. Lysosome fusion with the bacteria? No, the lysosome would fuse with the endosome where the bacteria is, right? Like once the bacteria is marked by the opsonin, that will facilitate the uptake of the bacteria in the endosome. That's endocytosis, will form the endosome, and then the lysosome that contains the enzymes will bind to the endosome, and that's where the phagolysosome will happen, and there will be the degradation of the bacteria. I wanna take your question, but I don't want to forget to mention something. The macrophages, even if there's no opsonization, they can still uptake bacteria. The opsonin makes it much better and much efficient, the process, okay? And it's typically necessary when you have an infection. Mm-hmm. Yes, please ask your question.
I just wanted to clarify, is endocytosis just the uptake of the bacteria, or is it the uptake of bacteria plus the forming that little bubble and partially killing it?
Is just bacteria or bacteria plus?
In the next lecture, there was something about it gets intaken and then it sort of forms a bubble around it and partially kills it, and then the lysosome fully kills it.
Okay. That's bubble around it is the membrane. So it's like the in cell biology, you must have learned about the endocytosis, the endocytic pathway. And basically, that's a pathway in which the cell will grab with its membrane, right, the bacteria or the microbe, and then that part of the cytoplasmic membrane becomes the vesicle or the bubble, as you said, where the microbe will be. And then that will bind with the lysosome, and that's called phagolysosome because it comes from phagocytosis in the case of the macrophage. And that's where the bacteria will be degraded. It's already in the endosome, there is low pH, and so it's possible that some impairment of bacterial replication happens there, but it's really in the phagolysosome where the bacteria is destroyed. Okay. Any question about that? Okay, let's do the next one. Thank you for your question. Okay, let's do the next one. Okay. Just a joke, eh? I enable reactions, and this is the reaction I got. I'm sorry, it's that early. Anyway, I thought that's funny. There is a question, yes. did you try A, C? A as an apple and C, my bad, I should correct here. AC, you tried, does it work? It works, okay, sorry about that. I should have corrected there, my bad. Okay, there you go. Okay, so let's stop this now. And so most of you answers C. How is the bacteria ultimately destroyed after the formation of the phagosome, fusion of lysosome with the phagosome, forming the phagolysosome, right? That's the correct one for sure, and almost all of you answered that one. And I'm still trying to figure out how to show you this, but anyway, so there you go. Okay, there you go. Okay, so, and the other A says release of complement proteins into the cytoplasm. That's definitely not true. The complement proteins are extracellular and they act extracellularly. Remember, they are synthesized by the liver, and then they are soluble proteins that are in the blood, in the plasma, in the extra fluids, remember? Okay. Opsonization by antibodies. That's not ultimately how it's destroy the pathogen. That's another way in which opsonization can happen, and we will learn about that. And that comes after the adaptive immunity, that mode of opsonization, right? But we learned there are different ways in which the bacteria can be opsonized, right? C3b is one of them. Antibodies is another one. Mannose lectin is another one that was in the recorded video. C-reacting protein is another one. Perforation of the bacteria membrane by C3b, that's not true because the one that initiate the perforation is C5b, and then that leads to continue other proteins cascades, and it's another complement, I think it's CC9, but anyway, I'm not asking you to remember more complement factors, but this macro complex that form in the membrane that leads to the lysis of the bacteria. And so that's not underlying or triggered by C3b. Okay, let's do the next one. Yes, please.
What initiates the cleavage of C5 to C5A to C5B?
That's a good question. C3b is the one that will be opsonized, and once C3, that's a good question. Let me remember. I'm blanking out on that now. C3a is the anaphylatoxin. I believe, and I could be wrong because I'm blanking out on this, but I believe it's C3, is downstream of C3. And I believe it's C3a, but I could be wrong, that initiate the C5 cleavage. Do you remember? Yeah, C3b is... Okay, sorry about that. So it's C3b, I didn't remember which of the two. So C3b initiates also the cleavage of C5 in C5b and C5a. And I know there are many names and it's easy to be confused. What I think that you want to make sure you understand is a cascade, right? That after C3 cleavage, that will trigger C5 cleavage, and so it will release this C5a, that is the anaphylatoxin, and C5b that initiates the cascade to form the spore in the membrane. So always alarm and destroy, right? Okay, thank you for your question. Okay, any other question? Okay, let's go with the next one. Okay, so I'll stop this one right now. And so most of you answer A, and then this, which of the following is not a characteristic of inflammation? And so we learn inflammation starts with increasing vascular dilation and increasing vascular permeability, right? So vessel dilation and increasing vascular permeability. Which mediator, which factors can induce inflammation? You have learned already. Guys. Cytokines, yes. TNF alpha, for example, and you learned that in the recorded video, yes. Exactly, right? Like the anaphylatoxin C5a and C3a can induce dilation and increase in vascular permeability. And that's what when the process is initiated, right? Like if you dilate, there is more space in between endothelial cells that allows and also there is more blood that gets to this vessel because it's broader now, and so that gets to redness because there is more blood, and then there is more space in between endothelial cells, and that gets to release or leakiness of extra fluid, right, plasma, as well as proteins, including complement proteins, and also it will be allowed extravasation of the cells because there will be chemokines that are released there. And C3a and C5a are chemokines, for example, as well. TNF alpha is not a chemokine, it's a cytokine, but TNF alpha can stimulate the endothelial cells for the endothelial cells to produce chemokines, and you learned that in the last video, and that will also favor neutrophils and other leukocytes to extravasate, okay? And so that causes the fluid, the proteins, and the cells cause the swelling, okay? And also make pressure in the nerve ends and cause the pain. And then last is the heat. Remember these 4 characteristics of inflammation, and heat is because it increases the amount of blood circulation in the area. Okay, cool. So, and then production of functional BCR is completely wrong. This, we haven't started now, but you will see this is the gene rearrangement that occurs for forming a BCR or a B-cell receptor, and this is something we will learn later. But that's, remember we mentioned that the adaptive receptors are encoded by genes that are generated in a lifetime during B-cell development or T-cell development by gene rearrangement. That's how we form a functional BCR, and we will learn about that. It's fun. It's a little bit challenging, but it's fun, okay? Okay, cool. Let's do the next one. We were just talking about that, so I want everyone to get it right, and I'm giving you 5 more seconds. Okay, I'll stop this one now. And so all of you actually answer the B, and so C3a and C5a increase vascular permeability. We just said that. And they will also stimulate migration or extravasation of immune cells because they are chemokines, okay? Okay, that's it for the question. Any question there? Okay, so let's do the next one. Okay, so 5 more seconds. Okay, I'll stop it now, and then almost all answer correctly. B, right? Like we were just talking, the increase in vascular permeability leads to increased fluid leakage from the vessels and extravasation of complement and plasma protein, right? and it will not reduce the fluid leakage, right? And it will not decrease complement activation. If something will promote, indirectly at least, complement activation, because there will be more complement proteins in the tissues where the pathogens are, and that can activate the alternative pathway. And it will itself, vascular permeability will not have an effect in migration of monocytes, but the same anaphylotoxins or indirectly cytokines that leads to vascular permeability can have directly or indirectly through endothelial cell activation can lead to migration of monocytes and neutrophils. Okay, any question about that? Okay, let's do the next one then. We're just talking about that, so 5 more seconds. Okay, I'll stop it now. And then almost all of you again got it right. Anaphylotoxins like C3A and C5A cause the classical sign of inflammation, and we're just talking about heat, redness, swelling, and pain, and what is behind each of them, right? Like that's what you want to know. Not only memorizing this one, but know what causes this, right? And then you also need to learn them in Latin. No, calor, rubor, tumor, and dolor. You may hear about that, but of course we will not ask you to remember that. Okay, that's the correct answer. What can I add to that? Itching is very interesting. I think itching is a sign, right? Like when when we have in some cases, like an infection or not an infection, but the exposure to a toxin or an allergen cause itching, right? And I think it's so fascinating, guys, because, and I only realized I became fascinated with itching very recently, because itching means that there is something in your skin, and we were always thinking about this as part of the immune response, right? Because it's to eliminate that toxin. That's the form in which you physically eliminate the toxin, right? And we do that, but the animals do that in an uncontrolled manner. When my dog has allergies, and so when he gets dermatitis and he starts itching to an extent that cannot stop, can destroy, and can even lead to death if it is not prevented and treated. And so it's super strong, the stimulation that can be, an organism can respond to that to an extent that will compromise its existence, right? And so it must be that toxin can also compromise the existence, and that's why we evolved a way to really try to eliminate it. And so we always thought, okay, eliminate the toxin, eliminate the helminth, because, we always thought itching is Th2 responses, which we'll learn later that is tailored to helminths, is part of the immune system. But there is a fantastic global immunotalk that was given by Caroline Stockel from Harvard. It was 2 weeks ago. You probably will not understand everything now, but she talked beautifully about how actually it's an interaction between the nervous system and the immune system. And so our sensory neurons, and this is emerging, right? It's not for a textbook yet, but sensory neurons will sense this toxin. Part of it is even unpublished. Sense this toxin and then tell your hand or your mouth, in the case of the animals, right, to go and do that movement and start eliminating this toxin to an extent that can compromise the life of the organism. I think it's fascinating, all the biology that is behind that for that to happen. And as I said, we are still not understanding. And this talk is really, I think, revolutionary because it changed the way in which we used to understand what causes itching. And anyway, so if you watch it, if you go globalimmunotalk.org, you will see the website. There is the way to connect to Zoom or to connect through a YouTube channel, Global Immunotalks, and you can find it. But yeah, I highly recommend it. So we learned about fever as well in the past recorded video, right? And we will talk about that, but that's a systemic effect of cytokines acting in the brown adipose tissue that is the one that regulates thermogenesis. Dizziness, I don't think that it has anything to do with the immune or defense, and not at all. So fatigue, it is. Many times when we feel sick, and we study a little bit that in the lab. So I'm also very fascinated with that because it talks about the interaction between the immune and nervous system. So fatigue is a sign, is a response to infection many times, right? Like you have experienced, like this lethargy. And we are studying that in the lab as we speak. A graduate student, Josh, can attest for that. And so we're trying to figure out which neurons are responsible for this lethargy in the context of a viral infection. And we have identified the neurons and we can manipulate the neurons. And it's all unpublished, and so I'm not going to share, not because I don't trust you, but we want to make sure that we communicate the absolutely right facts. But anyway, that fatigue is part of a response to an infection. It's part of what is called sickness behaviors that include this lethargy or fatigue, and it also includes anorexia many times, or anadipsia, okay? We talk about pain, we talk about fever. This one, in all honesty, I haven't heard. I imagine is when the pressure through the nerve endings is strong, that can lead to numbness. But anyway, that's it, what I have to comment on this. Any question? Okay, you guys dictate how you want the lecture to go a little bit, not 100% because we need to cover, but if you're curious about something, I'm happy to derail a little bit to answer and respect that you have woke up very early to come today. Anyway, so let's go to the next one. Okay, so I'm going to stop this one now, and then all of you answer B, and this is what we have talked about, right? These anaphylotoxins, which are C3A and C5A, they increase the migration of monocytes and neutrophils from the blood into the tissues because they act as chemokines as well, correct? Okay, let's do the next one. We haven't mentioned this with this name, the membrane attack complex, but this is the complex that the complement, the pore that the complement forms, right, that we mentioned. So the correct answer, and I will stop this one now, the correct answer is C here. It forms pores in the pathogen's membrane leading to cell lysis. So this is, again, we haven't learned it with this name, but all what I care mostly is that you know what it does. It will lead to destroying the pathogens through the formation of pores. Which other factors we have mentioned that leads to the formation of pore in the membrane of the pathogen? C5b will initiate this cascade, will not be the one forming the pore, will initiate the cascade of protease. So this C5b is a protease, and it will trigger cleavage of another protein, and activation of this other protein that is a protease, and so on. It will go until it leads to the formation of this complex, okay? But we learn about non-complement factors that are leading to the formation of pores in the membrane of pathogens. Do you remember? I don't even know what is. You know more than me. What is it?
I think it will get recruited by C5b.
Yeah. No, I'm talking no complements. No complement. No, I don't want you to remember all the name of all the complements. It's too much, and we need to. There are some names that I need to ask you to learn because it's like language. We need to communicate. But so I try to minimize that because I want to minimize memorization, and I want to stimulate you to understand why things happen, right? But there is another factor, and you can use the general name, not the specific name. You can say complement, right? In this case, not complement. But, I'm not trying to get the specific names. It's just, you can say cytokines, you can say chemokines, but this one, what we talk that form pores in the membranes of pathogens. Yeah, antimicrobial peptides. These are peptides that are released by epithelial cells and in response to pathogens, and they can form pores in the membrane of pathogens. Mm-hmm. Okay, microbacterial peptides also forms pores. Okay, so I think that with that we have completed the clickers from lecture 2, and so I will now project the lecture, the summary for lecture 2 and 3, and so we can summarize, and I can answer any question, the video that you watch for lecture 3. And so in lecture 2, we learn all about complement as a form of recognition, being complement soluble proteins. But in lecture 3, you learn how this recognition can also occur by cell-associated receptors that can be either in the surface of the cells or in the cytosol. And I guess that the best separation of these cells-associated receptors are the ones that are membrane-associated, because the membrane-associated can be in the surface, right? Or can be also in the vesicles, because there is membrane in the vesicle. Do we have toll-like receptors in the vesicles, in the endosome? Which ones are in the endosome? Do you remember? Yeah. 3, 7, and 9. And these ones in general, the one in the endosomes, and recognize nucleic acids. In general, I'm saying generalities here. And so CpG, toll-like receptor 9 recognize unmethylated CpG. Toll-like receptor 7 recognize single-stranded RNA, so nucleic acid. CpG, unmethylated, toll-like receptor 9, unmethylated CpG DNA. And then toll-like receptor 3, what does it recognize? We'll learn about that. Double-stranded RNA, that is intermediate in viral replication, okay? Which other factor, pathogen recognition receptor, recognize double-stranded RNA? The RIG-like receptors, right? Or CARDs, I think that's the other name we learned them with. So the RIG-like receptors where are they? Are in the membrane? The cytoplasm. And so let's, maybe this is helpful, right? Like we have the pathogen recognition receptors, and we have, can be soluble, and this is the complement, right? Or can be membrane associated. The membrane associated can be in the surface. Give me one example of the ones in the surface. TLR6 and TLR4, TLR 2, 4, 6, okay? And then we can have some that are in the membrane associated that are in vesicles. And then we just said that these are the ones that recognize the nucleic acids that are 3, 7, 9. TLR 3, 7, 9. And then we have the pathogen recognition, so the solubles, the membrane, solubles and extracellular, let's say. Extracellulars. And then we have the ones that are in the cytosol. Okay, so these two are associated with cells. One is in the membrane, that can be in the surface membrane or in the vesicles, and the other ones are in the cytosols. And we just have a Rig-like receptor. Rig-like receptors, right? Or the other name that we learn them with is the card receptor, card family of receptors. This is equal, we use it as a synonym. Anyone that you want to remember, card receptors. Okay, so that's an example. And then we said that these two, the Rig and the toll-like receptor 3 binds the same pump, right? The same pump, the same pathogen-associated molecular pattern that is double-stranded RNA, correct? Okay, now I want you to think and answer, share your thoughts. Why do you think that we have evolved to have more than one receptor pathogen that recognize the same pump? Yes. Yes, right? So the pathogens have evolved means to subvert and evade recognition and other aspects of immune defense. And, is it locked? Yeah, I don't know what. Oh, sure, we should make sure it's not locked. If you can help with that, just, we don't want to be locked. Okay, there. So just making sure for safety, we don't want to be locked, right? So I was saying, okay, so exactly. So you have pathogens evolve means to subvert the different immune pathways, including recognition. And so that's why in immunology, many times there is redundancy, right? Because the pathogen, for example, evolved to evade toll-like receptor 3, right? And so then the host evolved another PRR to recognize double-stranded RNA, and so can still recognize a pathogen that evolved to evade toll-like receptor 3 recognition. This is the type of redundancy that has been the product of this race between the pathogen and the host for millions of years of coevolution. Make sense? Okay, yes, and there was a question there.
I was just going to ask for clarification on the writing of it.
Yeah. Oh, sorry. This says pathogen recognition receptors, and trying to categorize them so it's easier to understand. These are soluble extracellular proteins, soluble, right? And that the one that we learned there is the complement. And then we have a membrane and cytosol. And then within the membrane associated, it's in the slides as well, within the membrane associated proteins, we have surface membrane associated proteins and vesicle membrane associated proteins. And then we have the pathogen recognition receptors that are in the cytosol. And that's, but also we learn other ones, not like receptors, right? That will trigger inflammasome, which is a kind of aggressive layer of activation. So many times, these ones that are in the surface or the ones that are in the vesicles are triggered first, and maybe triggered by commensals even that are not damaging, just to keep in check the commensals. And the ones that are in the cytosols are by pathogens that somehow subverted. They were not able to be eliminated in the vesicles. For example, some bacteria, as I said, are impairing their destruction in the vesicles, and then they can inject or replicate in the cytosol, right? And that is a dangerous bacteria, and that will lead to the activation of these cytosolic receptors. And in the case of the bacteria, the DNA of the bacteria will activate the NLR receptors that will lead to inflammasome. And again, that's inflammasome is a cascade of signaling cascade that will lead to activation of caspase-1 and cleavage. Again, is protease. Most of the signaling we learn is kinases, remember? But in the case of the inflammasome, the signaling cascade is protease and will lead to activation of interleukin-1 beta. That is one cytokine, and it's a potent cytokine that can be damaging. And so that makes sense if you think about it. We want to if you are fighting an enemy and you think that you know that there will always be some collateral damage, you want to start with, something, or you don't want to deploy your very distracted armament against an enemy that doesn't need that type of armament, because that will create unnecessary collateral damage, okay? So, and that makes sense with the immune system as well, right? That we don't deploy the whole armament and the most damaging one against our commensals, right? Okay. Anything, but you know that commensals can cause disease as well, right? Like we know, learn about Candida albicans. It can be kept in check, but we need to keep it in check. Otherwise, it can outgrow and colonize and cause disease, right? Like as it happened in HIV-infected patients. And so, commensals also require some innate response and adaptive response to some extent to keep them in check. Okay. Any question about these pathogen recognition receptors and what you learn in the lectures? Yes. Remember, I want to summarize this here. This is always recognizing a pump, right? This comes from the pathogen, okay? Whatever pathogen, can be bacteria, can be a virus, but that pump binding to the pathogen recognition receptor in the host, this is the pathogen, will lead to this recognition. And remember, this recognition then leads to signaling that basically in general are kinases, except for the exception I just mentioned, right? With the inflammasome, that is a protease cascade. But in general, it's kinases. And these kinases can lead to the activation of what? These transcription factors, right? And for example, we learned two transcription factors, NF-kappa beta So the pathogen recognition receptor binding to the PANT will get, there will always be an adapter here, an adapter that is necessary to activate the signaling. All this is in the slide and the recorded video. I'm just reviewing a little bit for you. That is necessary for the signaling. We learn for some of these pathogen recognition receptors that leads to NF-kappa beta activation. The IRAC, the IKK kinase is activated. That leads to phosphorylation of the inhibitor of NF-kappa beta. And this inhibitor is always associated with NF-kappa beta. So when the inhibitor is phosphorylated in response to this cascade, so now this inhibitor is degraded, and then NF-kappa beta, which is the transcription factor, can translocate into the nucleus and initiate a cascade of gene and protein transcription. So it initiates genes transcription and protein, and these proteins and genes, these proteins that are synthesized by this cell that saw the pathogen, what will be the ultimate goal of these proteins? To do what? Alarm and destroy, right? Alarm that are other that there is dangers in the body, and try to destroy the pathogen as much as possible, depending on how well equipped is the cells, right? If it is a macrophage, it's greatly equipped. If it is an epithelial cells, may not be that much. But, trying to destroy as much as possible, because you want to keep the pathogen in check. So when it comes the bigger armament, there are less infected cells, and it's easier to eradicate the pathogen without much tissue damage. And that sometimes will happen. And that's how many times is the immune system that kills the organism. And I don't want to derail too much, but for example, many times when with the respiratory infections like influenza or SARS-CoV-2, right? The problem is not that the immunity doesn't get there, it does just get too late. And so by the time it gets there, just the pathogen has spread a lot, and so the immune system trying to eradicate all these infected cells ultimately cause, disease and can cause death, okay? So there is always a timing issue, and that's why it's important to always try to destroy or contain pathogen replication and spread as much as possible early on. The other transcription factors we learn is the IRF, right, that leads to the production of interferon proteins. Okay? Any question? Okay, yes.
So, with the question from the video, like where you were asking to predict the consequence of TLR9 knockout and
Yes.
TLR7 and RIG1 knockout, so essentially those should be minimal just because there's overlap.
Depending on which pathogen is the one that infects. I think I have these clicker questions here. Let's, but yeah, those are good to. Let me finish summarizing these, and then we'll switch right to the clicker question. No, excellent. No, please. And if I didn't answer, you can ask in the office hours. Okay. Alarming other cells of tissues, and we learn there are different these cytokines. We said it will, or proteins. That some of these proteins are cytokines, will lead to alarming other cells and tissues. And these examples of that are cytokines which, like IL-6, that induce production of mannose binding protein or C-reactive proteins by hepatocytes, activation of other cells, and we learn how type 1 interferons can activate natural killer cells, and we learn how natural killer cells will act, destroying the pathogen, but also type 1 interferons can activate cell-intrinsic antiviral immunity, and we learn in different genes or interferon-stimulated genes that mediate that process. All that is in the video. Also, alarming will be through recruiting other cells, and we learn about chemokines that can do that, and also having systemic effects. One systemic effect is the interleukin-6 that is released by immune cells and has an effect in the liver. Another one is fever, for example. And so the other, and the last effect is destroying the microorganisms, and we just said, so preventing the spread as quickly as possible. We talk a lot about phagocytosis by macrophages and neutrophils. Natural killer cells are immune cells, innate immune cells, and they have granules that are released when they have engagement of an activating receptor, right? And without engagement of an inhibitory receptor. And are cytokines that can enhance their activity, like type 1 interferons. And then complement, we will talk a lot about that, can create these pores in the membranes of the pathogen and damage the membrane, and so they can do these antimicrobial peptides. Okay, any question about that? Yes?
I was just wondering, with the fever being part of the alarming, isn't the fever bring up body temperature so they can block the microbes' growth? Isn't that part of the destroying?
Excellent question. So the cytokines are the ones that will alarm by causing fever, I guess, that as a systemic effect. That's the way we think to study it. But you're right what you're saying, right? Like the cytokines will alarm, and one way they will alarm is they will interact with the brown adipose tissue to cause fever, increasing thermogenesis, and so it increases the heat in our body. And so, but that fever is aimed at destroying ultimately. The same thing that the cytokines will alarm by activating natural killer cells, right? And these natural killers are the ones that will destroy. Okay? Did it answer the question? Yes? Okay, cool. Any other question? Okay, let's do the clicker. Okay. So I'll start this one now. Okay, so I'm going to stop this one now. And then most of you answer C, and basically which of the following best describes pathogen-associated molecular patterns, and is molecules broadly shared by pathogens by distinguishable from host molecules, right? And that is very interesting because it can be distinguishable not just because of the structure of the pump, but also can be distinguishable because of the localization. So there is CpG DNA in our genome, unmethylated CpG DNA islands in our genome, but they are in the nucleus. They will not interact with toll-like receptor 9, which is in the vesicle, okay? And so when there is a pathogen that it gets, or bacteria that gets into the vesicles, that will lead to the interaction with toll-like receptor 9. And so it's not just that this distinguishable may be distinguishable because it's different structure, right? Like LPS, for example, we don't have LPS, okay? So it's distinguishable because it's a different structure, or maybe distinguishable because it's a rare structure in the cell, and it is in a different location, okay? So host molecules, no, they are pathogen-associated molecular patterns. unique molecules shared by different host cells. That's not true. Antibodies, that's not true. Okay, any question about that one? Okay, let's move to the next one. Okay, so I'll stop this one now. It's quite easy. We have talked about it. And most of you got it right. And so it's B, right? What is the primary function of a toll-like receptors? Is they recognize molecules broadly shared by pathogens known as pathogen-associated molecular patterns. So that's in the board right now. And so they produce antibodies? No, right? They directly destroy the pathogen? No, they don't directly destroy the pathogen. They will lead to a signaling cascade that will lead to transcription factor activation, that will gene transcription, protein synthesis, and these proteins may lead to alarm and may lead to destroying directly these proteins directly, or they may activate other cells like natural killer cells that destroy, or as we just mentioned, the brown adipose tissue that will create fever that will destroy, right? Okay, so yes, please.
Before we move on,
Yes.
I wanted more clarification on like how localization plays a role in PAMs.
Yeah.
So you said that because TLR9 is in the endosome or its vesicles, and then the PAMs are somewhere else, they don't have that interaction until there is a pathogen that creates that interaction.
Close. But thank you so much for asking the clarification. Yes, because I'm sure if you I didn't explain it well for you, there are other ones that have the same question. So the toll-like receptor 9, right? And I put this one as one example, is recognizing this CpG unmethylated DNA, right? It's not that we don't have CpG unmethylated DNA in parts of our genome. We do, right? And so then you have a pathogen recognition receptor that is recognizing a structure, CpG unmethylated DNA, that is in bacteria, is more abundant in bacteria, by the way, but it's also in our genome, and so it's not that it's unique to a pathogen, right? So now, maybe you can think why we are not all the time activating TLR9 in the cells, right? If we already have CpG and methylated DNA, why not? Why toll-like receptor 9 is not constantly being activated even without pathogen? That may be the purpose, right? You don't want that to happen, right? And indeed it doesn't happen. But why it doesn't happen if toll-like receptor 9 can recognize our own DNA? Why it's not activated? Right? And so where is our own DNA? In the cell. In the nucleus, right? Where is toll-like receptor 9? In the endosome. So that's why it's not constantly being recognized. So now when a pathogen is uptaken, it will expose part of the DNA in these vesicles, and now TLR9 can be activated, but only when the pathogen puts the DNA there, right? Like the CpG DNA there. Make sense? Yes? Okay, perfect. Any other question on that? Or any other question? Okay, cool. Let's do the next one then. Thank you for asking. Oops. Oh, this one is easy. Okay, let's 5 more seconds. Okay. So we just talk about, even in the board, we talk about toll-like receptor 4, which is a pathogen recognition receptor in the surface and the membrane in the cell, in the surface membrane of the cell, recognize lipopolysaccharide that is in all gram-negative bacteria. Okay, and this structure is not in the host, the lipopolysaccharide. And this, in the case of TLR4, is not sufficient. It needs to associate with MD2 and CD14 that form part of a complex that recognize this lipopolysaccharide to trigger the signal. Okay, can we move on to the next one? Yes? Okay, so let's do the next clicker. Okay, let's stop this one. And yeah, I can see how some, most of you answer A, which is the closest one, but none of the other. I think that it says secretion of cytokines. Definitely will be secretion of, okay, what is the ultimate outcome of the toll-like receptor 4 signaling pathway activation? Definitely will be secretion of cytokines, right? Because these proteins which include cytokines that leads to, and chemokines that leads to alarming, right? And activating other cells that destroy. However, what I don't like, and I can see how some of you may have answered none because of that, is that it says via the endoplasmic reticulum. And so the cytokines are synthesized in the endoplasmic reticulum, and then through the Golgi are then released. And so, I guess should say synthesis of cytokines via the endoplasmic reticulum. I know that it's not exact, the answer, and so maybe some of you, that's why you answer none. But anyway, so the closest one is A. Okay, the secretion of cytokines is the ultimate consequence. Okay? Next, yes?
So you said it should not be secretion via the ER, it should be secretion via the Golgi?
Yeah, but just the general cell biology, just the ER will participate in the synthesis of these cytokines, and then through the Golgi, but that's in general when when the cell secretes something, it would be through the Golgi apparatus, right? And that will lead to the secretion ultimately. So that's why it's not exact, right? The ER is involved in the synthesis of surface and secreted proteins, always the ER, but the Golgi is ultimately involved in the secretion. So, I don't know, if you want to be very picky, you should write via Golgi, okay? Or synthesis of cytokines via the endoplasmic reticulum. Maybe that's, yeah. Okay?
Yeah, it's just our slides actually have a diagram that says they're synthesized in the cytoplasm and secreted via the ER.
Yeah, I would argue that they are synthesized in the ER because surface membrane and secreted proteins are synthesized in the ER. Yeah. Okay. What else? And these are definitely secreted proteins. The cytokines will always be secreted. Other question? Okay. So let's do this one then. Okay, I'll stop this one in 5 seconds. Okay, let's stop it. And so the LPS, right, can activate 2 adapters, MYD88, that leads to signaling and activation of NF-kappa beta, and TRIF adapter that gets to signaling and activation of IRF transcription factor. And NF-kappa beta leads to induction of many proteins and synthesis of many proteins, including many cytokines that we call pro-inflammatory cytokines. And IRF leads to production of interferons, which are cytokines that we said we talk induce. They will be secreted, activate the transcription of interferon-stimulated genes. And almost every cell in our body has the receptor for interferon. So if we have release of interferon, then every cell in our body will be able to sense this alarm through the interferon and will activate these interferon-stimulated genes. There are hundreds of genes that create an intrinsic antiviral state, and we also learn that interferons can activate NK cells, and we'll learn other immune cells as well. Okay, so IRF leads to so the correct answer here is B. Okay, so let's do the next one. Okay, so let's stop it now. And so these, all of these pathogen recognitions leads to the activation of interferons through different adapters. So the RIG-I leads to production of interferon and IRF3 activation. Toll-like receptor 4, we just mentioned that. SIGAS is an enzyme, remember, that recognizes cytosolic DNA, and this leads also through another adapter to activation of IRF3. And all of these ones leads to activation of NF-kappa beta as well. I'm not asking you to learn all the adapters, but all of these leads to the activation of, through different signaling cascades, leads to activation of IRF and NF-kappa beta. Okay, so activation of interferons and other pro-inflammatory cytokines and chemokines such as TNF. Yes?
So is it just the adapter that determines whether it activates NF-kappa beta
Good.
or interferon?
Yes,
Okay.
yes. So they associate with different adapters, yes. Because there are experiments, for example, with TLR4. If you mutate the adapter TRIF, it will perfectly produce TNF, but will not produce interferon. If you mutate the adapter MYD88, it will induce interferon, but not TNF. Okay, any other question? Okay, so let's do the next one. So what is the prime? I'll stop this one now. What is the primary function of SIGAS in the cytosolic DNA sensing pathway? SIGAS, again, is a unique pathogen recognition receptor because it's an enzyme, right? Typically it's not. And so it will, so it says it acts as a transcription factor. No, it's a pathogen recognition receptor. It recognizes cytoplasmic DNA and catalyzes the production of SIGAMP. And this SIGAMP, this intermediate, guanosine adenosine monophosphate, this intermediate that is produced after SIGAS binds to the bacterial DNA is what will bind to the adapter. In this case, it's called STING, and will initiate the signaling to activate IRF and NF-kappa beta. Okay? It degrades its cytosolic DNA? No. It's a pathogen recognition receptor. It facilitates protein-protein interactions in the nucleus? No. Is it in the cytosol? Okay? Any question? Okay, so what I would like to do is do one more of these ones, and then the rest of the clicker questions will all be solved in the discussion sections that they start today, right? Today is the first day of discussion section with A4 group. Remember, before Thanksgiving, we have to start the week on Thursdays. And so today is the first day of discussion section for group A4. And so, but you know, everyone will have the chance. And so Shob and Eric and Kathy will be running different discussion sections. Shob will be running all of them, and Eric and Kathy participating. And so all the remaining clicker questions will be solved in discussion sections in preparation of midterm, and also, of course, during office hours with me and with the TAs, you can ask questions. Yes? No, because the discussion section's purpose is to have small groups, and so as it says in the syllabus, you have to attend the 1 year. If you have a problem, needs to be well justified and pre-approved, okay? Yeah, okay. So do you have time maybe with one? The graph on the left, let's do it, let's post it, and then let's do number 5. Okay, so let's stop it now. And so there are, remember, the TLR7 is the one that will recognize single-stranded RNA virus. So you will have no recognition, but you know, also all viruses generate double-stranded RNA. So RIG-I will be, and this one, you know, in all honesty, should say toll-like receptor 3 as well, because RIG-I recognize double-stranded RNA, and so you still have toll-like receptor 3. And so I think that you will have some innate response, and so probably B is correct, because there will be double-stranded RNA recognized by toll-like receptor 3. However, I think that if this said toll-like receptor 3 as well, number 5 is the one we are doing, right? Then the correct answer will be A. Is that clear? Okay, let's do quickly number 4. So remember, toll-like receptor 9 recognizes what? CpG DNA, right? This virus is an RNA virus. It's a single-stranded RNA virus you are infecting here with. So it cannot recognize CpG DNA, but it has toll-like receptor 7, it has toll-like receptor 3, it has RIG-I, right? Okay, so we'll end with this one then, and let's stop it now. And most of you answer C, which is the correct one. So you will still have recognition through toll-like receptor 7, toll-like receptor 3, RIG-I, right, of this virus. And so C is the correct one. Okay, so this, job, I think that we should revise all of these. Some of these are solved in the video, and the rest we have solved today, but I think that we should revise here and the rest of the clicker questions in section. And then we will start with adaptive immunity next week. And next week, start counting the clicker, okay? Okay, take care, guys. By the way, I have office hours, as it says in the syllabus. I start today with office hours at 9:30.